I would like to thank the organizers for giving us the opportunity to share a few insights about our programs at GenSight Biologics. So GenSight Biologics is a biotech company developing gene therapy in inherited retinal diseases leading to blindness. We have two main programs using two technology platforms. Our lead program is the development of a gene therapy in LHON. We have another program in retinitis pigmentosa, and we are at early stage for this program with a first-in-human study which is ongoing. GenSight was founded in 2012, and the headquarters is based in France. So I would like to focus this presentation on Lumivoq gene therapy development in LHON. Let's start with LHON disease. Before onset of the disease, the carriers of these mitochondrial mutations have a very good vision, 20 out of 20, and one day subjects have a dramatic drop of vision, first in one eye, and a few weeks later in the second eye. You see this drop of vision on the left graph. Rapidly during the acute phase of the disease, these subjects reach their lowest visual acuity, and it's called the nadir. And this is typical of this disease, a rapid and sequential drop of vision. And within 1 year, these patients become blind. Different mutations on the mitochondrial DNA can lead to LHON. The 11778 mutation on the ND4 mitochondrial gene is the most frequent mutation, representing around 75% of LHON in Europe and in the US. It is also the mutation causing the most severe clinical form of LHON, and Lumivoq specifically targets this mutation. This is a representation of what LHON subjects face during their daily life. Clinically, this manifests with a central vision loss, which is the hallmark of LHON. They get a central scotoma, which rapidly enlarges and becomes very dense, with a dramatic impact on their quality of life. So we developed technology of gene therapy to overcome genetic mutations in the mitochondrial DNA. The gene therapy Lumivoq specifically targets the 11778 mutation on the ND4 mitochondrial gene, and Lumivoq is a recombinant adenovirus vector type 2, containing the wild type, the healthy mitochondrial ND4 DNA. Lumivoq is injected via intravitreal injection, and it is the best way of administration to reach out the targeted cells, the retinal ganglion cells. Once injected in the eye, the ND4 DNA is transcribed into mRNA in the nucleus. Then the two mitochondrial targeting sequences actively shuttle the ND4 mRNA to the mitochondrial surface, where a functional ND4 protein is synthesized and translocated inside the mitochondria to restore energy production. So this is a unique technique that can be really extended to other mitochondrial mutations. And this gene therapy with this target on 11778 mutation on the ND4 mitochondrial gene is specifically treating these LHON subjects. So there are two phase 3 trials, reverse and rescue studies, which are pivotal studies which have been designed in close collaboration with the EMA. So these trials are completed. They have a similar design. They are randomized, double-masked, controlled studies aimed at comparing Lumivoque with the control. So each patient had one eye injected with Lumivoque and the other eye was injected with sham injections. So as you know, it's mimicking a real intravitreal injection, but no product is injected in the eye. The main difference between reverse and rescue is the timing of administration. In reverse, patients were treated when they had a vision loss between 6 months and 1 year, and in rescue, they were treated earlier when they had a vision loss within 6 months. So on this graph is represented the mean visual acuity over time for these two trials, for reverse and rescue. And they have a follow-up at 2 years post-injection. So in red, the treated eyes, and in blue, the sham eyes. And in solid line, reverse, and in the dotted lines, rescue. You see that the treated eyes and the sham eyes track together with similar curves of visual acuity improvement over time. And when looking at both reverse and rescue studies, you see that the improvement of visual acuity is totally coherent between the two trials, with the same shape of recovery across these two trials, evolving really in parallel, with a recovering time around the same time point when you consider the time from vision loss. So importantly, if you look at these curves again, the patients are moving from off-chart level of visual acuity to on-chart level of visual acuity when the treatment starts to be effective. So, because we actually lost, in a way we lost our control, because our primary endpoint was the difference of improvement of visual acuity between treated eyes and sham eyes. And you do understand that we have a bilateral improvement for treated and sham eyes. So the eyes tracking together, it was extremely important to compare these results with the natural history of the disease. That is why we have been running a natural history study called REALITY. This is a multi-center international observational study of LH subjects. And on this graph, you see the REALITY curve in solid green line, showing that these patients, when they are not treated, they get a dramatic visual acuity decrease, especially during the first year. And then there is a progressive decrease over time and over a period of 5 years with no recovery. And again, you see that the shapes of reverse and rescue trials, so for treated patients, have a totally different dynamic, showing a consistent visual recovery over time, totally different to the natural history curve represented with REALITY study. It is also interesting to look at the improvement of visual acuity, no more from baseline, but from nadir. Initially, we talked about the natural history, the disease, and that patients drop vision dramatically to reach a nadir. If you look in this trial, the improvement of these patients from nadir, this improvement is major, remarkable, because it reaches 28 letters for a reverse trial and 26 letters for rescue trial. So it means 28 letters, it's almost 6 lines of improvement, and 26 letters, it's more than 5 lines of improvement. So it's a huge improvement from Nadia. And again, it's far above the threshold of clinically meaningfulness, that is actually 3 lines of improvement. There are other ways to look at efficacy, and we looked into responder rates, and this is equally important to detail these results. So when you look at responder rates, and we took again the threshold of improvement of 15 letters from Nadir, responder rate being defined as having achieving this response in 1 or both eyes, you see that 76% of reverse subjects got this response and this improvement, and the responder rate for rescue was 71% of the subjects. And this improvement again means that firstly there is coherence and homogeneous response for the patients with a very high level of responder rate. And also it shows that these results are not driven by few outliers results. And this is important to show the global response in the trial. So, since these patients actually were treated unilaterally, and you saw that we got a bilateral improvement in the eyes of these patients, we decided to conduct a non-human primate study to better understand the mechanism of this bilateral effect in humans. So the objective of this non-human primate study was to evaluate the potential in vivo migration of Lumivoq active substance from the injected eye to the contralateral injected eye in these primates. So 3 monkeys were unilaterally injected with Lumivoq product with an equivalent dose of a human dose. And we looked 3 months after injection at the visual systems. And as expected, we found Lumivoq DNA in the injected eyes of these monkeys, and these are the green bars. But more importantly, we found out Lumivoq DNA in the contralateral non-injected eyes of these monkeys, so in the anterior segment, in the retina, in the optic nerve of these contralateral non-injected eyes. So with this experiment, we could demonstrate that there is a transfer of Lumivoq DNA from the injected eyes to the contralateral non-injected eyes of these non-human primates. So it gives a lot of insights why we are seeing this bilateral effect in clinical trials. Let's move on to safety. So we have a lot of experience with the product. In total, 189 patients were injected with Lumivoq. There were no serious adverse events in treated eyes. There were no discontinuation due to ocular events. The main ocular, actually the main ocular adverse event is intraocular inflammation, which is classical with gene therapy. But the good thing with Lumivoq is that it's mostly mild, and this intraocular inflammation is responsive to conventional treatment, usually to corticosteroid drops only, and it's resolutive without sequela. So we have a very good tolerance, and there is no systemic safety issues. And this is not surprising because the biodissemination, when injected in the eye, the biodissemination of the product is negligible. So next slide is about another trial, which is actually a phase 3 trial, which is ongoing. And this phase 3 trial has been closely designed with the FDA, and it is under a special protocol assessment. So the FDA requested to test the bilateral treatment of Lumivoq as HN is a bilateral disease, and to compare the visual outcome between eyes of different subjects. That is why the pivotal study design with the FDA is a bit different from reverse and rescue trials, which has been designed with EMA. So to move on to an important point, which is compassionate use. We have programs of compassionate use in the US and in Europe. So in the US, 4 patients were already treated bilaterally through compassionate use, and we have additional requests which will be shortly submitted to the FDA. In Europe, we have a compassionate use which is running in France through what we call a nominative ATU, authorization of temporary use. We have already 3 patients who were treated bilaterally through this compassionate use, and we have additional requests which have been submitted to the French agency. So in conclusion, as the regulatory pathway has been different between EMA and FDA, we'll be filing for EMA registration in September this month. Actually, we are currently filing for EMA. And this is based mainly on reverse and rescue pivotal studies, which has been agreed and designed with EMA. And we'll be filing for BLA, so to the FDA next year, so in Q3 2021, once we get actually the results of the ongoing pivotal study REFLECT, which has been designed with the FDA. That's why there is a bit of schedule in terms of regulatory pathways. I would like to thank the patients, the investigators, the study teams who have participated in these clinical studies. Thank you for your attention.